So exam Monday, this covers chapter 4. This is the one on protein production.
Stress.
Remember upstream, downstream, stuff like that. Column chromatography 5. There's microbial. Biotech. Chapter 6 is agro that we're on right now. Let's see, in addition, remember we have antibodies, Western, blot, and ELISA. I have done that this came after the last exam. So that will be on it. You guys should have all the slides. And then cell culture. Cell culture slides. Now this excludes hemocytometer and cell count. So excludes hemocytometer. Because hemocytometer is really falling under lab. Where cell culture, I gave you guys all the slides. Remember cell culture, we talked about primary cultures and how to keep cells sterile, aseptic, and hoods and stuff like that. So that's all part of cell culture. Any question? Normal format. So it'll be 50 multi-choice. Bring a pencil. And a Scantron form that I'll give you. And then, I don't know, somewhere around 12 short answer. As usual, some of the short answer to questions will be similar, if not identical, to ones you had on the quiz. Quiz is up from end of exam 1 to through today. Okay, counting today's quiz. All right? And these will total 50 points. They're roughly 4 each. And some of them are longer, and there'll be 5 points, maybe even a 6-pointer in there, or something that's really long. Okay? Any questions? I will give a short review at somewhere around 11:15. So stop lunch at 11:15. Give you guys a short review. If anyone has any questions on anything, please ask at 11:15-ish. Okay? Any questions other than anything on the exam or material? Questions about this stuff? Okay, several people have asked about lab. Lab is a swap, so we're going to run it in 2 phases, and each is 1 hour long. So you'll be swapped. One half of the lab, I believe, is the front lab, does mycoplasma with Dr. D in the main lab, and that is a PCR. And next week we run a gel. The other half does the bioreactor prep. And then we swap. It's designed to run about an hour each, so nobody should be paid. Okay, so my expectation is that most, if not all of you, will be gone by about 2:15, 2:30. Okay. Any questions? So So we were going over, obviously, you can see this exploding. So let me give you a big picture again. Can I erase this? Everybody okay? The big picture is kind of slide here.
And I know that that's, like I know it's chapter 4, chapter 5.
So, So big picture is when we talk about crops. Okay, there's a huge sort of a grassroots kind of back to nature, anti-GMO groundswell that's gripping the nation, in fact, much of the world. So there's a huge anti-GMO campaign being waged against basically modern biotechnology techniques as opposed to applied to agriculture. So this campaign is largely fostered out of ignorance, and what you guys should have learned so far is I'm empowering you with knowledge. So when you're at a a dance or a dinner or a bar or something, and this topic comes up, you can say, no, 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 we learned that this is all nonsense. So 2 key factors. Number 1, artificial selection. Right? Humans have been doing this for thousands of years. So the corn that you buy in the store today in no way looks like what the American Indians and the Chinese, etc., started with 9,000 years ago. Totally different, totally not recognizable. So this is all done by humans over thousands of years, just picking the best ears of corn and planting them each year. This gave rise to, remember, the double hybrid cross. Right? This is kind of the really sophisticated form of artificial selection. The double hybrid cross, remember, this was patented. This is the so-called hybrid vigor. Okay, so if you keep self-pollinating a group of crops, the productivity goes down, down, down. Plants get smaller, ears of corn, for example, get smaller, more prone to infections, etc. So double hybrid cross basically relies upon a feature called hybrid vigor. Okay? You guys should all know. 2, GMO. Genetically modified organisms. And this is what we do today. So basically, this process, enormously successful. Look at broccoli or any of the brassica, which no way resembles what we started thousands of years ago. GMO hugely accelerates this process. Okay, so because of GMO, you can basically in one generation produce a huge exponential improvement in some quality or some property of that particular crop. This is what this has been so successful. This was basically launched in the mid-90s. So GMO is the last phase of this. Okay? So if you look at hectares industrial development given to genetically engineered crops, you can see this is exploding upward. We went over the slide before. This is showing 1996, where there were basically 0 GMO crops planted, and this is skyrocketed up. We'll go through all these things. HT, BT is a particular toxin, etc. So we're going to go through all this stuff shortly. But you can see that percent of plant, this is percent now, not totally, but percent on many of these crops is approaching 80, 90, 95%. So GMO has basically taken over agriculture. Conventional selective breeding, hybridization, cloning, protoplast fusion, we stopped on this slide last week. Leaf fragment technique, I'm going to talk about next. Gene guns, chloroplast engineering, and antisense technology. So all this stuff's coming up. And these are ways basically of producing quantum improvements. You could suddenly in one generation produce a crop which has significantly better properties than what you see. So instead of waiting thousands of years, you can do this in one growing season. Conventional selective breeding and hybridization. This is the double hybrid cross that we went over. Sexual cross, 2 lines, repeated back cross until hybrid offspring and parent, until you get what you want. So in order to do this, obviously you're picking a parent that's nice and vigorous and grows well, and then you're taking some hybrid that, say, produces well, and you keep crossing them until you get the property that you want. It's a very slow process, takes years. Polyploid plants, multiple chromosomal sets greater than normal, very, very rare in humans, as everybody knows, and this usually leads to dramatic loss of function. For example, Down syndrome, which is a very serious neurological deficit. It's called mongolism. In plants, this is common. Okay, you do not get adverse effect. In fact, you often get plants with increased vigor, even though you wouldn't guess that doubling the number of chromosomes is a good thing to do. In many plants, you can double, triple, or quadruple them. Okay? Increase the desirable traits such as size, whole chromosomes transferred rather than single genes. Cloning, growing plants from a single cell. Protoplast. Protoplast, remember, anybody remember what is a protoplast? What is a protoplast? Anybody remember? Unless he's not sure. John?
Does it, it's a plant cell that like lacks the cell wall or something, it's like de-differentiated?
Perfect. So protoplast, no cell wall, it's been digested away. So the cell is basically spherical, it's alive, it's going to try to grow its wall back as fast as possible, but before it does that, it's basically like a mammalian cell that has a plasma membrane around it, hence the name protoplast. No wall. So it's easily amenable to all kinds of techniques, such as fusion, such as gene guns, etc. Protoplast cell is a callus cell whose wall has been dissolved by the enzyme cellulase. Fusion of two protoplast cells creates a cell that can grow into a hybrid plant. Examples include Brocco flower. So you make a protoplast cell from broccoli and another one from cauliflower, and you literally physically fuse the two cells together. This is not fertilization. It usually gives you double the number of chromosomes. Remember, broccoli and cauliflower, same genus, same species. It's the same plant. It just looks different. So you can fuse protoplasts and end up with something that's a hybrid of the two. So if you guys have seen this in the store, it's kind of a green broccoli-looking cauliflower. They're pretty good. Huh? Like Brocco flower, protoplast. So protoplast fusion with generation of transplanted plant. This gives you the steps. Chop the leaves, get the leaves and chop them up. Treat with cellulase, sugar and salt. Isolate protoplast by centrifugation. You spin out the garbage, you spin these guys out. They can be fused at this point to create hybrid plants, simply by mixing them together and getting them infused. Throw protoplasts up on filter paper over agar containing feeder cells, which is shown here. Feeder cells are also called nerve cells. So these are cells that are producing factors that the protoplast cells need in order to develop into a full plant. Okay, this is relatively easy to do with plants. Much harder with animals and much harder with mammals. Throw protoplasts on filter paper over agar containing feeder cells, transfer colonies to media high in its cytokinin. We'll talk more about cytokinin. This is not a cytokine. Cytokines is something totally different. Cytokinin is a plant hormone. We'll talk more about it later. And low in auxin, another plant hormone. So do not confuse cytokinin with a cytokine. Cytokine is a factor which gets cells to activate and move around. So for example, anaphylactic shock in people, which is often fatal, a person that is allergic to peanuts or bee stings can drop within seconds. Right? It's called a cytokine storm. There's sudden a rush of cytokines through the body. Cells all become activated, T cells, B cells, etc. They dump all the stuff out, neutrophils, etc. And a person basically has serious problems. Cytokinin is a plant growth hormone. It's much different. Transfer to root, inducing media, no cytokinin, no auxin. Transfer to soil, regeneration, you finally grow up this plant. Obviously, you have some very careful conditions so it doesn't get a fungal infection or something. Yeah, auxin, there are about a dozen or so plant hormones, and auxin is one of these plants that was discovered as critical. Gibberellins, auxins, etc. Which I'll talk about later. Leaf fragment technique. So that's protoplast fusion, right? This first method you guys have learned, protoplast fusion. Leaf fragment technique. And this dominated agrobiotech for a couple decades. This was the early technique. Small discs are cut from leaves, cultured in media containing genetically modified agrobacter. This is a bacterium that infects plants. And it causes something called a crown gall tumor. So if you look at, I should show a picture. Like you can go into your backyard or a park or something, and some big old elm or oak tree, and you'll see this huge sort of knobbly growth coming out of the side. That is a crown gall tumor caused by this bacterium. Okay? So agrobacter is Agrobacterium tumefaciens, the soil bacterium that infects plants. Bacterium contains this TI plasmid. I know it looks like an L, it's TI. TI plasmid. So method number 2 is the TI plasmid. This is from agrobacter or agrobacterium. Okay. This plasmid, just like all the plasmids we talk about, such as E. coli plasmids, etc., can be genetically modified using all the techniques you guys have learned so far this semester. So you can cut it with endonucleases, you can stick pieces in, and then you can use this to infect plants or these leaf cut discs. DNA from the TI plasmid integrates with DNA of host cell. Leaf discs are treated with plant hormones to stimulate root and shoot development, just like I showed in the previous slide. Generally auxin, cytokinin, sometimes gibberellins, etc. So this is showing the plant you wound it. This is the TI plasmid, which is from this agrobacter. And it is a plasmid, right, which is separate from the host DNA. This infects this wound and produces this tumor. So plant cell chromosome, tDNA integrates in the plant cell genome. So this plasmid, and in blue is some cool gene that you stuck in there, which you think is going to benefit this crop. That ends up being expressed. It's now in the plant chromosome. Okay? This is just like all the techniques we've gone through so far. This applies to plants. Plants, you have to use agrobacter. This is just like the story, because it affects plants. Producing transgenic plants. Good slide to review. Dissect leaves, chop them up. Put in recombinant agrobacteria containing this TI plasmid that you engineered and stick in them. Culture with feeder cells, transfer to shoot-producing media, usually auxin, gibberellin, cytokinin. Transfer to root-producing media, put growing. And this guy will now have integrated into its DNA present in every cell, whatever this gene is that you stuck in there. Got it? This is a classic transgenic, because you're putting something in there that's not normally there. Agrobacterium, key transgene vector. Right? It's like a little suitcase for getting genes in the plants. Mostly used for dicots. The world of plants is divided into monocots and dicots. Monocots tend to be things like grasses. It includes corn. Dicots are things with big, broad leaves that are not monocots. So this is the world of plants. Specifically, mainly monocots, cereals, maize, rice, sorghum, wheat are not amenable to agrobacterial infection. Okay, so this does not work with these plants. These are all angiosperms. This basically led to the development of the awesome gene gun. Gene gun allows you to basically introduce transgenes into both monocots and dicots. It's most successful for monocots because there's no other way to do it. You cannot use agrobacteria. So John Sanford at Cornell University introduced the gene gun, generating the first genetically altered corn plant, and this was done back in somewhere in the 1990s. Don't know how to explain it without giving a whole course, which is part of spring. So the world of plants, I have to do this just very quickly. So the world of plants obviously are divided into huge categories, which contains thousands of different species, each hundreds of thousands in the case of angiosperm. So gymnosperm, or naked seed, gymnosperms appeared first in the fossil record. These are a few 100 million years old. Gymnosperms include things like redwood, firs, pines, evergreens, etc. These are all gymnosperms. Okay, naked seeds, things with cones. Okay, angiosperms are awesome. Came out very recently, only about 150 million years ago. Angiosperms are what we're all used to. All of the kinds of good things to eat are angiosperms. Apples, pears, plums, tomatoes, anything that flowers, or corn. Angiosperms are split into two huge groups. Monocots, and this is, there's a whole 3 or 4 weeks on this in Bio 102 next month. Monocots and dicots. And the cot is referring to the number of cotyledons, which is a plant. Okay. That's all I can say. So just bear in mind that there are 2 different huge classes, and monocots are not amenable to agrobacterial infection. In fact, they're essentially totally resistant. The method does not work. Gene gun uses a .22 caliber cartridge, and it fires a tiny plastic bullet, which has a tungsten. W is the symbol in the periodic table for tungsten. Microparticles coated with DNA into an embryonic plant cell. Plastic bullet disintegrates, tungsten particles are released in the cell. You can fire this into specific regions of the cell. So these bullets are very small, much smaller than the cell itself. And the awesome thing, which came out only the last couple of decades, is you can fire it into the chloroplast. Now this means that you will only alter chloroplast DNA. Everybody remembers this is one of the great endosymbiotic events in the basically in the history of eukaryotic cells was this merging that occurred in chloroplasts, right? We had a cyanobacteria, some sort of green algae, that merged with some sort of cell, and you ended up with something able to harvest life. The chloroplasts are now free-swimming organelles. Okay, they have their own circular DNA, they have prokaryotic ribosomes, they synthesize their own proteins, etc., just like mitochondria, right? Everyone remembers this. Okay, chloroplast. So you can insert this into the chloroplast and not into the plant itself. Now this is good because whatever your transgene is never appears in pollen, right? Because pollen is just a reproductive grain with a host chromosome in it. There are no chloroplasts. So this allows you to avoid having to do all this government testing with your plant to be sure that wind-blown pollen with the transgene in it isn't going to cause horrors in some other farmer's field. Okay, so that's the idea. Use marker genes to distinguish genetically transformed cells. Same kind of thing that we went over with bacteria, where you can use antibacterial agents, blah, remember, beta-lactamase, which gives you penicillin resistance, etc. Antibiotic resistance. Technique is useful in plants, monocots that are resistant to agrobacter, where you have no other choice but to fire in a bullet. Here's a picture of the gene gun that fires a .22 bullet. Basically, it shows this cartridge, particles, some glass out, and it hits cells, and then you take these guys out, and you can fire it specifically into parts of cells, to the nucleus, vacuoles, etc., or chloroplasts. You grow them up, and you pick cells that have taken the gene up successfully by using selectively, like Eddie. Chloroplast engineering, you need DNA and chloroplast can accept from several genes at once. High percent of genes are inactive. DNA chloroplast is totally separate from DNA released in pollen. There is no chloroplast in a pollen grains are tiny. Okay, no chloroplast. No chance of transferring genes that get wind to distant crops. It hugely shortens the period of time that it takes you to go to market. Right? When you have to do wind testing, this takes a number of years. Transgenics. This is some mouse signal peptide, light chain cDNA, and heavy chain, and you cross these plants and you end up with something that now produces whatever it is that you want. So OVED introduced complementary genes into plants by recombinant DNA, such as gene gun or agrobacterium, cross plants by standard pollen transfer method, just like Gregor Mendel. People go around with a little brush and they snip anthers or the male pollen producing portions of the plant off of the recipient acre. New method introduced multiple genes into chloroplast. You fire them in with a gun. Chloroplast DNA is not transferred between plants. So it only gives you expression in that one plant. Okay, we'll be get rid of that. So number 3 is gene gun. Right, which will work technically on any type of plant, but particularly a monocot, which is not amenable to agrobacterium. Right, this only will infect dicots. Gene gun, subset here is chloroplast. And you can fire a transgene right into the chloroplast, and then use special promoters and all that's expressed in the chloroplast. Okay, number 4 is antisense. Antisense is something that should be somewhat familiar to you guys, I hope. Okay, if you will remember, a huge percentage of our DNA is transcribed and is not made into mRNA. So it is not transcribed and translated into protein or a gene product, correct? A lot of it does not fall into that category. A huge percentage of the RNA that we make is never made into proteins. It's all involved in regulation or control. MicroRNAs, which we went over, Dicer, this whole system, RISC, silencing system, there's all this type of technology. So antisense, what you do is you make a short piece of DNA that is complementary, RNA, that is complementary. In other words, it has the complementary sequence, so it forms a hybrid, to mRNA that you want to silence. Okay? So you can do this engineering. So you can make this short little 20-mer of RNA, induce it to a plant or get it to be expressed, and it will silence whatever gene you pick. And remember, just like CRISPR, a 20-mer is absolutely unique. Remember, the likelihood of the 21-mer occurring at random in a given genome is 0.01% or something. So the likelihood that you're going to knock out something else is virtually very low. So to insert complementary copy gene into a cell, gene encodes mRNA molecule called antisense. It's the exact opposite complement of the mRNA that you want to knock down. Antisense molecules find the normal mRNA, inactivates it, and this has been done to shut down certain genes that don't want to express. Why don't we stop there? We'll take a 10-minute break and reconvene at 10 o'clock.
Your lab
Fun. Gang, gang.
Okay, antisense.
That's
So
how it was with my dad. He had
example,
one at like 40-something.
So again, the way
Good.
antisense works is
What?
that
He's
know
good
the
now.
genes
He's alive, but he lost
then
20% of his
they,
heart.
in
And I
most
don't want
crop
that to happen
plants,
to me.
The genome is known, sequence is known.
Good Lord, but you also
So you can silence
you said
genes that you don't want to be active in order to get designed property. So for example, the flavor saver tomato,
There were 85 cents.
Design. called flavor saver. So here's a normal tomato. An antisense was introduced that basically the normal sense produces a protein that essentially causes the fruit, the tomato, to rot. Rotting is a desirable property because a rotting fruit usually tastes good, animals eat it, and then they excrete or pass the seeds out at a distance, and this enables propagation. So this is how angiosperms spread, either in the feces or droppings of birds, animals, etc. This is a common mechanism. So many fruit are designed to break down polysaccharides, produce sugars, make them sweet, and ultimately have them rot. So antisense is used to suppress this polygalacturonase, which is this PG enzyme, which catalyzes natural plant tissue rotting, and by inhibiting this, you prolong the shelf life, so the tomato rots much more slowly. This first GMO plant commercially approved. So here's this PG, the polygalacturonase gene, is sense mRNA antisense was designed. This was introduced, and basically it reduces the expression of PG, polygalacturonase, which is what's leading to the breakdown of these polymers and as part of the rotting process. Okay, so the last one, a huge percentage of fruit rots and is never consumed. Vaccines, genetic pesticides, herbicide resistance, enhanced nutrition, pharmaceuticals to fuel. So all these are the directions for GMO plants today, and all these different kinds of industries that have arisen. Vaccine is encoded in plants' DNA, for example, a gene from tobacco mosaic virus. This is another vehicle used for getting genes into, for example, tobacco plants. Protein produced from viral genes stimulates the plant's immune system, and the plant becomes invulnerable to virus. So if it is not affected by this virus, it lasts longer. Generate plant resistance to virus. TMB co-protein is converted to cDNA and is inserted into a TI plasmid vector. Remember, this is agrobacterium. Transfer to agrobacter vector, infect plant cells. This is being done for tobacco, which is the icon. Transform plant cell growth culture, regeneration now resistant to TMB infection. Okay, BT. BT is probably one of the biggest of the GMO genes. It comes from Bacillus thuringiensis. This is a bacterium that produces a protein that kills insects and their larvae. It literally chops up their GI tract, so they lose gastric integrity, and the insect dies. This has been used as a natural pesticide for 50 years, and what we have done is we've introduced this BT toxin into GMO plants. So now when caterpillars eat the plant or start eating it, they immediately die from consumption of this toxin. Inserted in the plant's DNA creates a built-in defense against certain insects. Crystalline form of the Cry-G protein breaks down cementing substance in the lining, breaks down the substance in the lining of the digestive tract of insects. Cry stands for crystallizing, so it actually produces a protein that forms shards that rip up the gut. Controversy surrounding monarch butterflies, but much data has been done to determine if monarch butterflies are being killed by this. As many people know, monarch butterflies become endangered. And are they affected by this process? And to date, the answer appears to be no. Everything all set on? Okay. Dr. D texted me, and she's going to come at around 12. This shows BT, naturally expressing cry gene product. Insect larvae normally eat treated plants. Plants expressing recombinant cry, and these caterpillars will die. BT bacteria naturally express cry. Insects are just going to. This is showing chi protein attaches to insect's gut wall, or breaks it down. Insect larvae eats plants that are sprayed, produce this BT, and they end up dying literally from the inside out. Safe storage. Millions of dollars are lost every year to insect infestation of crops during storage. Bless you. Transgenic corn that expresses avidin, highly resistant to pest and storage. Avidin blocks availability of biotin, vitamin required by insects to grow. Herbicide resistance. Traditional weed killers kill desirable plants along with weeds, right? So Roundup is the one we're going to talk about. Roundup is awesome, but the problem with Roundup is it kills everybody. So what you do is you use Roundup, which kills all plants, and you introduce Roundup resistance in crops. Now this means you can send a crop duster over a field of corn instead of weeding it, and all the weeds will die. The crop plant will survive. Okay. Somewhat controversial. Traditional weed killers kill everybody. Genetically engineer allows farmers to control weeds with chemicals that are milder and more environmentally friendly than typical herbicides. Roundup is this enzyme EPSPS, shikimate phosphate. This is involved in nucleic acid biosynthesis. So EPSPS is an enzyme, and glyphosate is Roundup, the active ingredient Roundup. It's called glyphosate. Bless you.
Thank you.
Right, and you can clear a field in a matter of a week or two. It'll be just bare dirt. Everybody dies. Crops transgenically produce an alternate enzyme are not affected by this glyphosate. Most soybean plants grow today contain herbicide-resistant genes. Unfortunately, glyphosate resistance obviously has evolved, which you would predict. So you're going to get somebody is going to appear with resistance. So this is a TI plasmid with this glyphosate insensitive EPSPS, so it's an altered form of this enzyme that is not sensitive to glyphosate, the active ingredient of Roundup. Produce this plant, spray everybody with glyphosate, and the transgenic plant does not die. Gene stack. Conventional selective breeding from two lines for genetically inserted transgenics, herbicide tolerance, and insect resistance. Plus, you can now produce plants that have both. So they can express this cry gene product. Remember, kills insects by ingestion and perforation of GON. And an EPSPS alternate is glyphosate resistant. And here's a Rhode Island farmer who's out in the Slocum area that I've talked to, just west of URI on Route 138. Purchases GMO corn expressing both Bt toxin and glyphosate. Remember, glyphosate now insensitive EPSPS. So this is not destroyed by Roundup. He drilled plants right into a field of wheat. So he takes this field, no clearing, no treatment, no nothing. Just plants corn right in this. Right? This is heresy. For hundreds of years, you always plow it, and then you plant sow corn, and then you start weeding it, or you crop dust it. Drill seeds right in. Corn sprouts, sprays with Roundup. Everybody dies except the corn. So this field looks like this. Okay, and all you do is spray with Roundup. Sprays Roundup, always die, only GMO corn survives. And he's reduced labor, reduced costs, reduced increased profit. The vitamin A deficiency kills millions of people a year, causes blindness, mostly in children. 4 genes from daffodil. I have a slide coming up. This is so-called golden rice. Golden rice? Anybody here golden rice? Good. Golden rice. Daffodil, and actually bacteria as well, that make beta-carotene. Beta-carotene is this long polyunsaturated molecule. It's a fat precursor to vitamin A, used gene gun and recombinant agrobacterium were used in order to produce this. It's called golden rice. Both genes and promoters of daffodil and Erwinia, which is a bacterium, insert these into agrobacterium, right? Transgene means it's not normally found in this bacterium. This is into the TI plasmid. In fact, rice cultures with TG agrobacterium grow gall plants, cross rice suited to various parts of the earth. And this is a key concept which you guys should take away. Although rice is rice, there are strain differences which are optimized for growth in the different areas. For example, rice you might grow in California is different from the rice plant, the strain of rice plant that you would grow in, say, Thailand. So strain differences have to be taken into account, because rice of a certain strain will do well in some areas and not so well in other areas. So basically, you have to make these GMO crosses against all these different types of strains so that it can be marketed around the world. This cute little slide basically shows you how to make golden rice. So Erwinia and daffodils, the genes were removed. These were stuck into TI plasmid using classic molecular biology techniques that you guys all know, right? And endonucleases to chop this thing, get it in, ligate it up, etc. Get this into agrobacterium, use this to infect the plant. So these genes give golden rice the ability to make beta-carotene by the introduction of a series of different genes. And the endosperm is the seed kernel part of rice that you eat. Along with promoters inserted plasmids, basically containing transgene, out of the petri dish containing embryos. As they infect the embryos, they transfer genes encoding for these things. Transgenic rice now must be crossed with strains of rice that are designed to grow in a particular area. So that introduces the transgene, making rice, into rice plants that can be grown around the world. So strain differences are a key concept. This is just showing latex plants. X plants, this is like a primary tissue culture where you're removing part of an organ, or in this case, part of a leaf. I don't think we do this next semester anymore, but you grow callus tissue, which are these sort of light greeny yellow blobs, and these are largely dedifferentiated, or essentially stem cells, which is our extension. And from these you can now grow whole plants. We actually had a lab in bio which we did, but we're not able to do this. So tissue cultures of sorghum plants, callus to shoots, and again you can see callus tissue forming from chopped up bits, and then these grow into plants and finally produce an entire mature plant. And this is normal rice versus golden rice. Golden rice now expresses these 3 genes that produce beta-carotene. Okay? A huge percentage of the world relies on rice as a major form of nutrition. Golden rice has been engineered to contain large amounts of beta-carotene. Body converts to vitamin A. However, as of 2011, no farmers planted golden rice due to concerns voiced by environmental organizations. Blocking the implement of golden rice has led to the deaths of millions of people in consequence, because basically this technique is available, and is available for consumption. CRISPR. CRISPR, remember, is a version of like the silencing, RISC, that I described, the RNA-induced silencing complex. CRISPR allows you to both silence genes. It essentially is a gene editing tool, so it allows you to make a cut at a specific spot that you designate based on sequence at some location within the genome. It also allows you to do gene insertions. So with CRISPR, you can insert genes uniquely into the chromosome in a given spot. This is incredibly powerful. This has basically revolutionized molecular biology in the last decade or so, since its inception. Altered promoter sequence control number of locules or seed compartments and fruit size. These are not GMO since you haven't done anything. All you're doing is silencing given genes during development. This is gene silencing. Remember, CRISPR is this guide RNA, which is the short little piece. It's about 21 minutes long. And Cas9 is this nuclease that cuts. So it cuts genomic DNA. Non-homologous ends joining in a piece is either missing or can be inserted. So CRISPR can do both, deletions and insertions of genomic DNA. So this shows the guide RNA cutting, and now you introduce new DNA, which has homologous ends to what's present in the DNA in a chromosome, right? In a chromosome of whatever the plant is, or a human, or anything, bacteria. So recombination, which is basically repairing, introduces this new piece into the genome. This allows you a very quick way to insert transgenes into plants and animals using CRISPR. Okay, the Arctic apple is another gene silencing of polyphenol oxidase, which basically causes apples to brown. Everybody knows you cut an apple, and after a couple of days, it looks like this. If you have the Arctic apple, it looks like this. Browns much more slowly simply by blocking polyphenol oxidase, which converts to heterochromatin. No new gene is introduced, so this is not considered GMO, because you're just silencing genes that are already there. The innate potato, again, is polyphenol oxidase being blocked, converting to heterochromatin. Heterochromatin is this intractable form of eukaryotic DNA where it's tightly wrapped around histone complexes and hence not available for transcription. So this is a way of silencing genes. This is also part of epigenetics, which we talked about. Produces less acrylamide, mutagenic with pride, also non-GMO, because all you're doing is knocking out genes that are there. So BT crops protect against insect damage and reduce pesticide use because you're killing the insect so-called naturally from within, producing this crying protein. Herbicide tolerant, such as the glyphosate, resistance to Roundup, disease resistance against, we discussed about tobacco mosaic virus, high performance cooking oils, healthier oils, delayed ripening, vegetables, increased solids, and tomatoes, which we talked about the locules, nutritionally enhanced foods such as golden rice. Just gives you do not memorize all these, but just have a feeling for them. Basically, every type of crop that you can think of has some GMO variant. And this harkens back to the slide I showed with the percentage of GMO crops available around the world, and it is skyrocketing. Most countries now do this, and it's up over about 90-85% for most crops. Basically, almost has some type of GMO variant. The future plant biotech pharmacology plants can be ideal protein factories. As I said in an earlier lecture, for example, treating people in Africa, tropical Africa, it's very difficult to store drugs. Refrigeration is expensive or unavailable, but by having the drug produced in a plant, you can then just transport seeds, grow plants, and then people eat plants in order to obtain medicines. So inexpensive edible vaccines do not require refrigeration. Molecular farming of phytochemicals, introduce chemicals useful in human health. Phytochemicals, which are just chemicals produced in plants. Antibodies, blood products, cytokines, growth factors, etc. Fuels. Biofuels, as everybody knows, when you pump gas into your car today, every pump will say contains 10 or 15% ethanol. The ethanol is produced from biologics. So this is from corn or something that's being broken down by bacteria in order to produce ethanol, which is then obtained from that. Usually waste products of crops. Need for alternatives to fossil fuels is increasing. It takes 7 gallons of gasoline to produce 10 gallons of corn kernel ethanol. So it looks like it's the thin end of the wedge. There's not much gain. So the amount of processing that's involved almost is equal to what you get out. I know it looks bad, but still there's a small gain. In the future, want to convert plant waste such as husks, stems, and sugars convert to ethanol. Algae may be the next alternative to petroleum. Okay, now comes the darker side or the lighter side, depending upon how you think. Health and environmental concerns. Anti-GMO, right, is an enormous movement. Say no to GMOs, and these are people marching on the Capitol, et cetera, in order to ban use of GMOs. Here's one of my favorite quotes from Norman Borlaug, died recently. American agronomists and humanitarian vet initiatives worldwide have contributed to extensive increases in agricultural production termed the Green Revolution. Received Nobel Peace Prize, Presidential Medal of Freedom, Congressional Gold Medal. Borlaug's goal, his mission in life, is to try to reduce starvation. The only way to do it, as everybody here should know, we are currently using 125% approximately of Earth's capable output, right? And we're using more than 100% based on fossil fuel consumption, overfishing, lumbering, all kinds of illegal types of activities, etc. So we are doing this in order to keep maintaining our population as today. Obviously, you cannot keep doing this, and the population is continuing to grow. So Borlaug was one of the ones that stepped in and said, we need plants that are going to be able to produce more. So each hectare or acre of plants has to give higher yield, or we cannot do this. Now, some of you in the audience, I'm sure, are saying, well, you know, to hell with it. We're just going to let all these people die and still eliminate GMOs. Well, you cannot do this. So Borlaug was not this type of person. What can we do to try to help? So received all this stuff. So at some conference, a heckler said, what do you think of GMO plants? And Borlaug's response is, fine. If you have a better idea, let's hear it. Right now, this is what we fix, 125%. The growing population, still exponential growth of human population. Okay, so how are we going to do it? Already, roughly a third of the world's population is undernourished or malnourished. You have to do something. Develop new strains of crops which yielded 4 times as much food per acre or per hectare, said to have saved the lives of over 1 billion people, making one of the most influential men in human history. Food is the moral right of all who are born into this world. Almost certainly, however, the first essential component of social justice is adequate food for all mankind. Civilization as known today could not have evolved, nor can it survive without an adequate food supply. And all of this, everything, every one of us sitting here today relies on artificial selection, right? And today, GMO, 90+% of everything you eat is genetically modified. So human health, what types of concerns have been raised? Opponents fear the effects of foreign gene bits of DNA not naturally found in plants. These can cause allergic reactions, antibiotic resistance, markers could spread to bacteria in humans, cause cancer. Okay, none of this has been supported by these concerns. Bear in mind, another concern I've heard from students in past lectures is, well, cannot DNA transfer from stuff we eat, to our cells? This is called horizontal gene transfer, and it has only occurred a few times in history of all life on Earth. It's an extremely rare event for genes to transfer from one organism to another, unless we physically do it, right, through CRISPR technology or something like that. So trans genes we can do very efficiently. But for this to happen naturally, it's extremely rare. There's a handful of cases, for example, of aphids that have ingested plant genes that allow them to change their color and hence become camouflaged on certain plant sources. So there are a few cases like that, but it's very, very rare now. 2,000 potato processing plants stopped buying GMO potatoes that were engineered to be pest resistant. However, there's no sign that they are inferior or dangerous. In fact, according to Borlau, they are essential, right, for the continuation of our society. Energy policy calls for 7.5 billion gallons of ethanol. I know it's only a few percent. You're gaining, what, 10 gallons for every 7 that it costs. But still, it's a gain of 3. To be added to American fuel supply by 2012. Balance indicates additional resources, mostly production-wise, for distribution of insecticides, needed to utilize traditional potatoes versus using GM potatoes in order to try to meet this need. Genes for pest or herbicides could spread to weeds. Few experts predict this will happen. Further studies are needed. There's extensive regulation, so studies are done. You cannot just suddenly start selling a GMO seed to farmers. It has to go through the USDA, United States Department of Agriculture, and it has to be approved before you can do this. So FDA and USDA oversees growing practices, regulates foods on market, environmental protection agency controls use of BT proteins and other pesticides. So all of these groups have basically said the safety of GMO is supported by all of these scientific groups plus medical bodies, etc. Ordinary tomatoes do not contain genes while genetically modified ones do. Is this statement true or false? Ordinary tomatoes do not contain genes. Okay, 45% of the U.S. population says yes, this is true. Only GMO plants contain genes. Obviously, it's just a misunderstanding. I mean, without genes, life is basically not possible. By eating a GMO fruit, a person's genes could be changed. This is, as I said, it's happened a few times in history of life on Earth. 9% of the U.S. says yes, public fears transfer of transgene antibiotic genes into humans. Horizontal gene transfer is rare. We eat plants all the time. And as far as I know, there are no green people that are able to absorb sunlight today. This is another one of my favorite characters. This is Patrick Moore. In his younger years, he was a very strong activist of Greenpeace and opposed GMO plants. And this is him in his younger years, and here he is today. Amazing what a suit and tie will do when, I guess, 20, 30 years of age. He now basically left this group and is in support of GMO, for which he has been vilified as an eco-Judas, right? Because he's turned his back and supporting GMO now, the evil agricultural company. Stop the Patrick Moore quote. The campaign of fear now being waged against GM is based largely on fantasy and complete lack of respect for science and logic, such as the questions I just cited. Right? A big percentage of the U.S. population thinks you can get genes by eating plants from them, and that GM plants contain genes and others don't. And the real balance is good. The real benefits of GM far outweigh the hypothetical and sometimes contrived risks claimed by detractors. Food is something that is taken for granted by most of the world leaders, despite the fact that more than half of the population of the world is hungry, and roughly a third is actually undernourished. Green side of agrobiotech. Biotech increases yield per acre, protecting millions of wildlife habitat, i.e. you don't have to clear as much land because you're producing more from the wildlife you have. Agrobiotech presents the greatest environmental achievement in history, higher yields of GMO plants, reduced soil erosion, etc., because you don't have to have as much farmland. Huge mega farms exist today. 3 huge conglomerate businesses in the northern hemisphere own the entire world seed market. Bayer, DuPont, and Chem China recently bought Syngenta. So Bayer purchased Monsanto for $68 billion. So these gigantic super companies are basically controlling the world's seed market, hybrid as well as GMO. DuPont purchased Pioneer Hybrid, now owns most of the old soybean, cotton, and corn. Merged with Dow Agro, and Chem China bought Syngenta for $43 billion in February 2016. Public distrust of science is huge. DDT, Silent Spring, remember this was Rachel Carlson's book that came out in the 60s, in which DDT, which was a common pesticide sprayed on plants, was found to feminize males and reduce amphibian populations, etc. Exxon Valdez, this was the oil spill in Alaska. Remember, this was a ship, an oil tanker that broke up and oil distributed, killing millions of birds. Bhopal, this Union Carbide pesticide leak kills 3,700 people. Chernobyl, Love Canal, Hooker Chemical Company toxic dump into Niagara Falls. BSE, this is mad cow. Tainted beef, mad cow disease, Creutzfeldt-Jakob in humans, 177 deaths. So humans that eat this beef can end up with these infectious prions. Remember prions? In last week's section. Tainted Coca-Cola, romaine lettuce, and it goes on and on and on and on. Romaine lettuce regularly gets pulled off the shelves. Basic fear of technology, unhealthy, unsafe, unnatural, high GMO data from public, increased herbicide pesticide use, farmers' environment, does not increase yields, and is starving the world. So these are all totally unsubstantiated. Long terms of GMO consumption, about 20 years of data generated worldwide. None show significant difference to non-GMO. None show health-related risks. GMOs are arguably more precise than conventional breeding methods because only known, precisely characterized genes are being picked. Right, the beauty of biotech is that you can take this gene out of this organism and stick it into this plant, and you're just changing that one thing. To our knowledge, every claim of a negative consequence to health or environment from use of GMO crops has failed to withstand scrutiny. The alternative to GMO inclusion in Africa is starvation, which is what Borlaug has said repeatedly in lectures. Right? We have no choice. If you guys can think of something, state laws given rise to federal law mandates that GMO be placed on packages. If you guys buy potato chips, almost all of them will say, no, this product contains GMO material. And consumers have the right to know. The problem is once you say something has GMO on it, everybody assumes it's bad for you. And so without education and without an understanding of what GMO is, by simply labeling, you're not really helping them. This is my fellow colleague, Albert Couch, who's down in Kingston campus, URI professor, and a tremendous agrobiotechnician, a very good person to know. Mandatory labeling alone is a poor substitute for education, conveys negativity to important agricultural teams. A label will not serve to inform the consumer, but rather misinforms the public that GMOs are to be avoided.
So with Monsanto, have you explained that they're completely down when they're putting pesticides into the field because they don't obviously want those pesticides to get into their skin, and then say that that isn't going to enter your body and eat that?
Okay, well, let's put this in there. Okay. What you're thinking of is Monsanto produces Roundup. And Roundup is glyphosate. And glyphosate is a small chemical molecule. This is not a gene. Right? So this is not gene alteration at all. So you're spraying a chemical compound onto fields. And glyphosate enters plants and basically knocks out the ability to make nucleic acids, and they die. So that's different because obviously human sensitivity to glyphosate is very low, but it is finite. And there have been many cases of cancers, et cetera, from crews which are inhaling, when you're spraying from a truck, there's an enormous amount of fog spray going out that workers are inhaling and probably ingesting as well. So that's why they're downed up. So that is different, right? You understand, it has nothing to do with GMO. That just has to do with with spraying crops with a chemical compound that's designed to kill weeds.
So how do you ensure that that isn't on the food that's brought to market?
How's that? That's actually controlled, and we go over it not in this class, unfortunately. So a lot of work is done in order to determine the types of levels that are present in plants when they're being harvested and for human consumption. So plants can be tested to see how much of these different herbicides and pesticides that are sprayed on that they contain. But bear in mind that the questions that you're raising is something that's been done to crops for decades before GMO, right? Crop duster planes have flown over since the 40s, spraying with all kinds of stuff, including DDT, which led to the silent spring. So there's no doubt that crop dusting and adding in foreign chemicals into the atmosphere as well as the plants we consume has a certain risk associated with it, just like the Monsanto lawsuits that are running by all the workers who are coming down with cancer. But that's different. That's not GMO based.
Right.
Right? GMO is just altering genes in order to give the plant some sort of benefit. No, it's a good question. The answer is it's all controlled. So when you buy an ear of corn, it's tested to be sure that it does not have more than a specific amount of phosphate and all these other agents in it. Because a lot of the effects on humans are either not known or is tested very little. So organic food debate. I just threw this in here. Dr. Couch talks about this as well. Organic food, 3 main requirements. So everybody knows what I'm talking about. You can go to the store and you can buy conventionally grown produce, or you can buy organic food, right? And anybody I like buying organic food. It often comes from local farmers, and it's usually much more expensive. But it's in this store, and it usually says organically grown. And organically grown has the following criteria. Farmer uses only organic fertilizer and not chemical fertilizer, such as ammonium nitrate. This is considered inorganic and not natural. Okay? Where do I talk about Haber-Bosch? Haber-Bosch. Have I mentioned the Haber-Bosch reaction? I don't think I have. It's Bosch. Oh, it was the probably single most important. This comes up in bio in the spring. So Haber-Bosch. You guys have to write this down because I don't have a slide on it. Oh, Bosch. Reaction. One of the most influential reactions that was ever designed, and this occurred around the turn of the last century. To do this, you need huge temperatures, huge pressure, and metal catalysts. Why is this reaction so important? Because what limits plant growth on this planet is nitrogen. It's generally nitrogen. Nitrogen, N2, is a gas that we all breathe in. In fact, 80% of air that you breathe in, every breath you take or every yawn you make, you're breathing in N2 gas. Almost totally unreactive. Very hard to fix this into a form that can be used by organisms such as plants. So this reaction enables you to convert atmospheric nitrogen, these are both gases. Haber-Bosch reaction. This is a gas. This is a gas. And it produces ammonia. And this can be oxidized to nitrate. is ammonia. And this is nitrate. Both of these forms can be used by plants. This cannot. So once this Haber-Bosch reaction occurred, it allowed us to fertilize fields and increase yields. Remember the Dust Bowl of the Midwest, right? Which led to starvation and abandonment in communities and suffering. So this reaction produces this, ammonium nitrate. This is not considered a normal, organic, natural fertilizer. Whereas malorganite, for example, is human excrement that has been dried and heated, and it can be used to treat lawns, for example, and fertilizer. This is considered organic, and this is not. So if natural pesticides are used, such as copper sulfate or sulfur, these are broadly toxic, and they're not considered more or less natural. And no GMO. When you buy an organic food, you are not buying GMO. Everyone understand? Sorry? Metal catalyst. You need a metal surface for this reaction to occur. Sorry. Is organic food better? Published literature lacks strong evidence that organic foods are significantly more nutritious. This is Lauren's question. There's a slight increase in pesticide concentration for conventional food or organic, but still well within allowable limits. So foods are tested to see how much glyphosate, et cetera, is in there. Haley?
I have a very silly question. The last slide, you said that the organic fertilizer was made from human feces.
Yeah.
The people that are against GMOs, if that person ate a GMO and then that fertilizer was used, isn't it technically in their eyes not organic?
Okay, say it all again. Not quite.
So, if the organic fertilizer is made by human feces, and that human that the feces came from ate a GMO, wouldn't they technically assume that it's not organic? Because GMOs can be
Haley,
transferred.
incredible. It's possible. Whatever you do, don't suggest it,
Okay.
because somebody's going to decide it's true.
Okay.
So what she's saying is it possible that the feces that are coming from people who have eaten GMO crops now have GMO genes? Obviously, from a scientific point, to the physician, it's absurd. So published literature lacks a slight increase in pesticide concentration of conventional food. Head of Organic Trade Association concludes nutritional quality between organic and conventional is essentially the same. Organic food standards are not relevant or irrelevant to safety. Organic food has broad appeal, but cost tempered demand. So a typical cost comparison is about 30 to 50% more, depending upon the store you go to. Organic food is more expensive, mostly because of labor and less efficiency. Historically, growing organic cotton required slave labor. Must crop 2 times more land to feed humanity from organic fields, which is the whole point of GMO. And this is just giving you a price list, typical price comparison for organic versus conventional food taken in the local market. Organic food does not mean it is safe. Recent outbreaks of foodborne illnesses have been linked to organically grown foods such as spinach, cantaloupe, green onions, sprouts, etc. So organically grown food is just as likely to be contaminated as conventionally grown. Basis of U.S. consumer preference for organic food. Many of us support local farmers, concerned about toxins such as pesticides. U.S. consumer preference for conventional food. Most people say it's too expensive. And there's more conventional food available. Sustainable agriculture. Water use, tillage, synthetic pesticides, little or low or no fertilizer, achieve higher crop yields, decrease food waste. 26% of food purchased in this nation is thrown out. 1996, first GMO crops, soybean, maize, rape, cotton appeared. 2010, about 148 million hectares planted GMO in countries, mostly America and Asia. GMO crop impact agronomically, environmentally positive, claim negative impact, not evidence, significant yield increase, a beneficial halo effect. Halo effect is something you guys should be aware of. decrease in pesticide use in non-GMO plots adjacent to GMO. Simply because of the fact that in the GMO plot, you have reduced numbers of insects and weeds. So because of that, plots nearby that are not GMO show benefit, right? Because you just don't have as many bugs and don't have as much stuff around as you do without GMO plants. Reduced carbon footprint, less energy consumed, recognition of pest and weed resistance to GMO. That is the end of this chapter. Why don't we take a 10-minute break and we'll reconvene in 5 minutes.